In this video, we are going to learn about micturition and micturition reflex. In this figure, we could see parts of excretory system in human female and male. Ureters are shown. Along with that, ureteric openings, then internal sphincter, then urinogenital diaphragm including external sphincter along with urethra region. In case of male, we could see detrusor muscles that is also present in females. Then a bone-shaped structure called a trigin. Then in case of male, we could see prostate gland is shown. Along with that, bulbourethral gland is there. So the structures are shown. And where are the external urethral openings shown in case of female and male are very important. What is micturition? Micturition is the process of emptying of bladder, also called voiding. We know that. Then structures involved in micturition: urinary bladder, that include detrusor muscles associated with that. Then urethra or sphincter muscle. Then parasympathetic nerves supply urinary bladder and urethra. Then what is the mechanism of micturition? In infants, accumulation of urine in the bladder activates stretch receptors in the bladder wall, generating sensory or afferent impulses that are transmitted to the spinal cord, where a spinal reflex is initiated. This stimulates involuntary contraction of the detrusor muscle and relaxation of the internal urethral sphincter, and that expels urine from the bladder. This is micturition or voiding of urine. So in adult, micturition occurs when detrusor muscle contracts and there is reflex relaxation of internal sphincter and voluntary relaxation of external sphincter. So the given picture depicts micturition process. On the left side, you could see a bladder which is full. Along with that, external urethral orifice is there. Then internal urethral sphincter is there. Then external urethral sphincter is there. Along with that, nerve endings sensitive to stretch are associated with that structure. Then spinal cord is connected with this bladder. So there are some reflex control happening in case of this micturition process, when conscious effort cannot override the reflex action. Then we could see schematic representation of this, where the red arrow represents nerve impulses to spinal cord. Then blue one represents nerve impulses stimulate contraction of detrusor muscle. Then there are green arrow which represent nerve impulses stimulates relaxation of internal urethral sphincter. Then next, right side figure indicate control of micturition after bladder control is established. So brain regions are involved in this process, like brain cerebral cortex region. Along with that, spinal cord, voluntary inhibition of reflex is happening. Then nerve impulses to cerebral cortex is associated with that. Then nerve endings sensitive to stretch, which are connected with this. Like that, there are some nerves which travel to spinal cord. Urethra is there, external urethral sphincter is there. Nerve impulses stimulate contraction of detrusor muscle. Nerve impulses stimulate relaxation of internal urethral sphincter muscle. Then conscious contraction of external urethral sphincter overrides reflex micturition. So that's how control of micturition after bladder control is established. So micturition is the process by which the urinary bladder empties when it becomes filled. The micturition reflex is the basic cause of micturition. So micturition reflex is the basic cause of micturition. This involves two main steps. First, the bladder fills progressively until the tension in its walls rises above a threshold level. Then second step, which is a nervous reflex called the micturition reflex, that empties the bladder. What is micturition reflex? As the bladder fills, many superimposed micturition contractions begin to appear. They are the result of a stretch reflex initiated by sensory stretch receptors in the bladder wall, especially by the receptors in the posterior urethra, when this area begins to fill with urine at the higher bladder pressure. Then sensory signals from the bladder stretch receptors are conducted to the sacral segments of the cord through the pelvic nerves and then reflected back again to the bladder through the parasympathetic nerve fibers by way of these same nerves. The Micturition reflex is an autonomic spinal cord reflex. It can also be inhibited or facilitated by centers in the cerebral cortex or brainstem. Once a Micturition reflex begins, it is self-regenerative. That is, initial contraction of the bladder activates the stretch receptor to cause a greater increase in sensory impulses from the bladder and posterior urethra, which causes a further increase in reflex contraction of the bladder. Thus, the cycle is repeated again and again until the bladder has reached a strong degree of contraction. Then, after a few seconds to more than a minute, the self-regenerative reflex begins to fatigue and the regenerative cycle of the Micturition reflex ceases, permitting the bladder to relax. Along with that, the Micturition reflex is a single complete cycle of progressive and rapid increase of pressure, a period of sustained pressure, return of the pressure to the basal tone of the bladder. Once a Micturition reflex has occurred, but has not succeeded in emptying the bladder, the nerve elements in this reflex usually remain in an inhibited state for a few minutes to 1 hour or more before another Micturition reflex occurs. As the bladder becomes more and more filled, Micturition reflex occurs more and more often and more powerfully. When the bladder is only partially filled, this Micturition contractions usually relax spontaneously after a fraction of a minute. The detrusor muscles stop contracting and pressure falls back to the baseline. Then, as the bladder continues to fill, the Micturition reflex becomes more frequent and cause greater contractions of the detrusor muscle. Once the Micturition reflex becomes powerful enough, it causes another reflex which passes through the pudendal nerve to the external sphincter to inhibit it. If this inhibition is more potent in the brain than the voluntary constrictor signals to the external sphincter, urination will occur. If not, urination will not occur until the bladder fills still further and the Micturition reflex becomes more powerful. Then this picture shows normal cystometrogram showing also acute pressure waves, dashed spikes caused by Micturition reflexes. Micturition contractions are shown. Basal cystometrogram is there. Then intravesical pressure, centimeters of water, is shown in the y-axis. The higher centers normally exert final control of Micturition as follows. The higher centers keep the Micturition reflex partially inhibited except when Micturition is desired. The higher centers can prevent Micturition even if the Micturition reflex occurs by tonic contraction of the external bladder sphincter until a convenient time presents itself. When it is time to urinate, the cortical centers can facilitate the sacral Micturition centers to help initiate a Micturition reflex and at the same time inhibit the external urinary sphincter so that urination can occur. Then here we could see ureter, body region, trigone bone, then bladder neck is there, posterior urethra, then external sphincter is also shown. So sympathetic nerves are associated with this and parasympathetic nerves also initiate some connections, then pudendal nerves connect with this. So ureter region is there, then body region is there. Into this region, this parasympathetic nerves and sympathetic nerves connect. But along with that, trigone region also connect sympathetics and parasympathetic nerves. But bladder neck only connects with parasympathetics, posterior urethra region. Pudendal nerve connect with external sphincter. Pudendal nerve only connect with external sphincter. Then facilitation or inhibition of Micturition by the brain. The Micturition reflex is an autonomic spinal cord reflex, but it can be inhibited or facilitated by Centers in the brain. The centers include strong facilitative and inhibitory centers in the brainstem located mainly in the pons. Several factors located in the cerebral cortex that are mainly inhibitory but can become excitatory. Then voluntary urination is only initiated in the following way. First, a person voluntarily contracts his or her abdominal muscles, which increase the pressure in the bladder and allows extra urine to enter the bladder neck and posterior urethra under pressure, thus stretching their walls. Then this stimulates the stretch receptors, which excites the Micturition reflex and simultaneously inhibits the external urethral sphincter. Ordinarily, all the urine will be emptied with rarely more than 5 to 10 milliliters left in the bladder. There are some abnormalities of micturition like overflow incontinence. The sensory nerve fibers from the bladder to the spinal cord are destroyed, thereby preventing transmission of stretch signals from the bladder. When this happens, a person loses bladder control or atonic bladder happens. Then instead of emptying periodically, the bladder fills to capacity and overflows a few drops at a time through the urethra. Then automatic bladder caused by spinal cord damage above the sacral region. If the spinal cord is damaged above the sacral region but the sacral cord segments are still intact, typical micturition reflex can still occur. Then periodic but unannounced bladder emptying occurs. Then uninhibited neurogenic bladder caused by lack of inhibitory signals from the brain results in frequent and relatively uncontrolled micturition. This condition derives from partial damage in the spinal cord or the brainstem that interrupts most of the inhibitory signals. Therefore, facilitative impulses passing continuously down the cord keep the sacral centers so excitable that even a small quantity of urine elicits an uncontrollable micturition reflex, thereby promoting frequent urination. Next topic, structure of urinary bladder. So urinary bladder is smooth muscle chamber. Two main parts are there, the body and the neck. So this picture we have already seen. Sympathetic nerves are connected to the ureter and body region and into the trigon, with parasympathetic nerves are connected with body, ureter, trigon, along with that bladder, neck. But pudendal nerves only connect with external sphincter. So ureter, body, trigon, bladder, neck or posterior urethra, then external sphincter, these are the regions. Then UB is made up of UB in the sense urinary bladder. Urinary bladder is made up of smooth muscles called detressor muscles. When contracted, these muscles can increase the pressure of the bladder up to 40 to 60 mm of Hg. Contractions of these muscles is major step in emptying the UB. There is a low resistance electrical pathways exist from one muscle cell to another. Therefore, an action potential can spread throughout the muscles from one cell to the next to cause the contraction of the entire UB at once. Then on the posterior UB wall above bladder neck, there is a triangular area called trigon. Then two ureters enter the bladder at the outermost angles of the trigon. Each ureter enters the bladder obliquely enough the detressor muscles and then passes another 1 to 2 centimeters beneath the bladder mucosa before emptying into the bladder. Lower apex, the trigon opens into the posterior urethra. The bladder neck is 2 to 3 centimeters long and its wall composed of detressor muscles interlaced with a large amount of elastic tissue. The muscles in this area is called internal sphincter. Its natural tone normally keeps the bladder, neck and posterior urethra for empty urine. Then beyond the posterior urethra, a urethra passes through the urinogenital diaphragm, which contains a layer of muscles called external sphincter made up of voluntary skeletal muscles. Then external sphincter muscles is under voluntary control and can be consciously prevent urination even when involuntary controls are attempting to empty the bladder. Then Innervation of bladder. Principal nerve supply of bladder is by the way of pelvic nerves, which connect the spinal cord through the sacral plexus. Pelvic nerves are of two types, sensory and motor nerve fibers. Sensory direct the degree of stretch in the bladder. Motor, parasympathetic nerve fibers terminate of ganglionic cells located in bladder wall. Short postganglionic then innervate the detrusor muscles. Skeletal motor fibers via the pudendal nerves to the external sphincter to control the voluntary skeletal muscles of the sphincter. Then parasympathetic innervation through hypogastric stimulate the blood vessels of the bladder vessel. This picture which we have already seen include regions like ureters, ureteric openings, internal sphincter, urinogenital diaphragm including external sphincter, urethra, detrusor muscle, trigon, prostate gland in case of male, bulbourethral gland in case of male, then external urethral opening. Next topic, urine formation. Clear amber colored fluid of slight acidic reaction excreted by kidney is urine. pH around 6, normal range 4.5 to 8. Color light yellow, volume excreted 1 to 1.5 liters per day. The volume depends on amount of water intake, environmental temperature, then diet, mental state. Odor is faintly aromatic, then specific gravity 1.020 into 1.030. Composition of urine, water about 96%, urea 2%, uric acid, creatinine, ammonia, sodium, potassium, chlorides, phosphates, sulfates, oxalates. Everything consists of about 2%. Then urine formation, process of blood cleansing. We know that urine is formed by filtering the blood at renal corpuscle and reabsorption of necessary substances in renal tubule. Mechanism of urine formation involve 3 steps like glomerular filtration, tubular reabsorption, along with that tubular secretion. Glomerular filtration, tubular reabsorption and tubular secretion. So what are the steps in urine formation? First, afferent arteriole is there, glomerular capillaries are there, efferent arteriole is there, cortical radiate artery, glomerular capsule, rest of renal tubule containing filtrate, then peritubular capillary to cortical radiate vein. There are 3 major renal processes like glomerular filtration. Look at the rose colored arrow portion which is happening in the glomerulus. Then tubular reabsorption, the whole red colored portions like peritubular capillary is associated with that process. Then tubular secretion, which is also happening in the peritubular capillary. So these are the steps involved in urine formation. First process is the filtration. We know that, like glomerular filtration. Blood pressure forces small molecules from the glomerulus to the capsule. Then filtrates glucose, amino acids, uric acid, urea. Here we could see a picture of glomerulus. Glomerulus is there, H2O passes out, urea also passes out, glucose, amino acids, uric acid, salts. Glomerular capsule is also there. So glomerular filtration process takes place through semi-permeable membrane of glomerulus and glomerular capsule. Water and large number of small molecules pass through. Blood cells, plasma protein and other large molecules are unable to filter to remain in the capillaries. They remain in the capillaries. Water and large number of small molecules pass through, but blood cells, plasma protein and other large molecules are unable to filter, so they remain in the capillaries. Then filtration is assisted by the differences between blood pressure in glomerulus and pressure of filtrate in glomerular capsule. Then blood flows into the glomerulus with the help of afferent arteriole. Then blood flows out of the glomerulus through efferent arteriole. Like Bowman's capsule is there, proximal tubule is there, filtrate is oozing out into that portion. Because the efferent arteriole is narrower than the afferent arteriole. A capillary hydrostatic pressure of about 55 mmHg builds up in the glomerulus. This pressure is opposed by the osmotic pressure of the blood, provided mainly by plasma proteins about 30 mmHg, and by filtrate hydrostatic pressure of about 15 mmHg in the glomerular capsule. So the net filtration pressure is therefore glomerular filtration pressure, capillary hydrostatic pressure minus osmotic pressure of blood plus filtrate hydrostatic pressure. Like GFP is equal to 55 minus 30 plus 15 gives 10 mmHg. Glomerular capsule is there. Glomerular blood hydrostatic pressure, which is represented by this rose color, which is exerting into the glomerulus. Then 10 mmHg. But blood colloid osmotic pressure, which acts upward. Then capsular hydrostatic pressure 15 mmHg, that is also acting upward. Then net outward pressure is 10 mmHg, which can be calculated with the help of subtracting capillary hydrostatic pressure minus osmotic pressure of blood plus filtrate hydrostatic pressure. For ultrafiltration, we need to know few facts and few terms like fenestration, then basement membrane, podocytes, pedicels, and filtration slits. First, fenestration. The endothelial cells or glomerulus have small pores, 50 to 100 mm in diameter. They are called the fenestrations. It cannot pass the blood cells. So, fenestrations are small pores of glomerulus or endothelial cells. Then basement membrane. The base where glomerulus lie. They are made up of extracellular materials like fibrils. They restrict the entry of proteins and large molecules. Then podocytes are there. Bowman's capsule is made up of two layers. The inner layer is made up of highly modified squamous epithelial cells. These are called the podocytes. Pedicels. Thousands of foot-like structures extend from each podocyte is called the pedicels. Then filtration slits. These are the spaces between the pedicels. This act as slits to filter the material. Generally, they filtrate medium-sized particles. Podocyte is shown, like endothelial fenestration is shown with the help of green color. Then glycocalyx region with the help of yellow color. Then glomerular basement membrane with the help of light red color. Then urine is formed in the capillary space. Endothelial cell is there. Slit diaphragm is there. Mesothelial cell, mesanglial cell, food process, etc. are shown. Then GFR or glomerular filtration rate. Volume of filtrate formed by both kidney in 1 minute is GFR. GFR is equal to 125 ml per minute, like 180 liters. 180 liters. Most of the filtrate is reabsorbed. Less than 1%, about 1 to 1.5 liter is excreted as urine. The next phase, tubular reabsorption or selective reabsorption, process by which composition and volume of glomerular filtrate is altered during passage through renal tubule. Reabsorb into blood those filtrate constituents needed by the body to maintain fluid and electrolyte balance and pH balance. Reabsorption is achieved by active transport, passive transport, osmosis and diffusion. Some constituents of glomerular filtrate do not appear normally in urine because they are completely reabsorbed unless they are present in blood in excess quantities, like PCT, glucose, amino acid, vitamin, hormone, calcium and some urea, PCT, LH and DCT, 99% of water, Na plus, Cl minus ions. What is PCT? Proximal convoluted tubule. It generally follows active transport. Firstly, it starts in PCT, which has microvilli and brush border enzymes. Basic substances which get reabsorbed in PCT are glucose about 100%, amino acids 100%, Na plus 70%, K plus 75%, Cl minus 50%, urea 50%, water 65%, HCO3 minus about 10%. Loop of Henle. Descending loop of Henle is selectively permeable only to water. Ascending loop of Henle is impermeable only for water. Then that region is permeable for Na plus, K plus, Cl minus. Then DCT help in reabsorption of urea, Na plus, K plus, Cl minus, HCO3 minus. Collecting duct reabsorbs air, water. This picture shows portions and their reabsorption mechanism. Glucose, amino acids, potassium, sodium, proteins, vitamins, lactate, then urea, uric acid, H2O, HCO3 minus, etc. are absorbed with the help of PCT. Then some other regions are also shown. Blood in capillary, filtrate in tubule, selective reabsorption from tubule to blood. Then renal threshold or transport maximum. Kidney maximum capacity for reabsorption of a substance. If the level rises above the transport medium maximum, the substance will appear in urine. Then hormones regulating reabsorption. Parathyroid hormone and calcitonin regulate reabsorption of calcium and phosphate. Then antidiuretic hormone or ADH increases permeability of DCT and collecting tubule, increasing water reabsorption. Then aldosterone increases reabsorption of sodium and excretion of potassium. Then atrial natriuretic peptide ANP inhibits NaCl reabsorption. So parathyroid hormone, calcitonin, antidiuretic hormone, aldosterone are atrial natriuretic peptide ANP. Then proximal convoluted tubular PCT reabsorption. PCT is the most active in reabsorption. All glucose, lactate and amino acids, most Na plus, H2O, HCO3 minus, Cl minus and K plus, 65% of Na plus and H2O, 90% of HCO3, 50% Cl minus, 55% Ka plus, etc. are involved. Then PCT, major site of reabsorption, 65%. Like proximal tubule is there, isoosmotic region is there. Proximal tubule, Na plus, Cl minus, HCO3 minus, K plus, H2O, glucose, amino acids, etc. flows out. H plus organic acids and bases travel into the isoosmotic portion. Why is PCT a major reabsorption site? PCT epithelial cells have extensive brush border. PCT epithelial cells have extensive number of carrier proteins. PCT epithelial cells have large number of mitochondria to support active transport. Then PCT reabsorption about 65% filtered Na and water and are reabsorbed by the PCT. Then most of the filtered Cl is also are reabsorbed in PCT. Then secretion. PCT is an important site for secretion of organic acids and bases like bile salts, oxalate, urate and catecholamines. Then tubular reabsorption. For reabsorption, substance must be transported across the tubular epithelial membranes into interstitial fluid. Routes of transport. Intracellular path is there. Water and solutes can be transported through cell. Then paracellular path is there, like through the junctional spaces between the cells. Then through the interstitium back into the blood, like ultrafiltration, bulk flow is happening. Then tubular reabsorption. Solutes are transported through the cells, intracellular route by diffusion or active transport, or between the cells, paracellular route by diffusion. Water is reabsorbed through the cells and between the tubular cells by osmosis. Then route of water and solute reabsorption. Filtration, lumen, excretion, reabsorption, peritubular capillary region, blood, paracellular path, transcellular path, solutes, passive diffusion process, active transport, bulk flow, osmosis, etc. can be seen. Then next process, tubular secretion. Substances not required and foreign materials like drugs including penicillin and aspirin may not be cleared from the blood by filtration because of short time it remains in the glomerulus. Such substances are cleared by secretion into the convoluted tubule and excreted from body in urine. Tubular secretion of hydrogen ion is important in maintaining homeostasis of blood pH. Then tubular secretion. Tubular secretion means secretion of waste from the blood into formed urine so that this waste can be eliminated. Then secretion of happening in the glandular cells are shown. H plus NH3 plus creatinine, uric acid, drugs, etc. are PCT relate. Then DCT secrete HCO3 minus NH4 plus K plus H plus. These wastes are taken up by the glandular cells of PCT and DCT from the peritubular capillaries. The waste materials come from peritubular capillaries and gets mixed into the nephritic filtrate in PCT. Glomerular capillaries are there: filtration, reabsorption, secretion and excretion. Bowman's capsule is there. Filtration process, where is that filtration process occurring? In the Bowman's capsule, right? In the glomerular capillary region, glomerulus region. Then reabsorption, peritubular capillaries are there. Renal vein, urinary excretion is happening in the collecting duct. We know proximal convoluted tubule position, then distal convoluted tubule position, then collecting duct region, cortex, outer medulla and inner medulla regions. Then nephron is there: filtration, reabsorption, secretion and reabsorption of water. Tubular reabsorption, return of filtrates from blood at the proximal tubule through diffusion and active transport. Proximal convoluted tubule is involved in that. Then tubular secretion, movement of molecules from blood into the distal convoluted tubule. Molecules, drugs and toxins, distal convoluted tubule. Then reabsorption of water, return of H2O via osmosis along the loop of Henle and collecting duct. Thank you for listening.